All right, welcome everybody. Today it is my pleasure to introduce Dr. Keesha Roach as our GGI seminar speaker. So Dr. Roach is a new addition to the UTHSC team. She just joined the College of Nursing as an assistant professor in the Department of Health Promotion, Disease Prevention in August of this year. The focus of Dr. Roach's research is on chronic pain, with a special emphasis on sickle cell disease and the characterization of pain phenotypes, biomarkers, and health disparities in individuals of African ancestry. She also participates in other areas of research, including vulvodynia, osteoarthritis, and cancer. Over her career, Dr. Roach has received training both at the NIH and the Pasteur Institute in France, and has degrees in both psychology and nursing. In 2018, she completed a PhD in nursing at the University of Illinois at Chicago College of Nursing, and then completed an in-part integrative and multidisciplinary pain and aging research training T32 postdoctoral fellowship with the University of Florida's Pain Research and Intervention Center of Excellence. After that, she was appointed as an assistant professor at the University of Florida College of Nursing in the Biobehavioral Nursing Science Department. Dr. Roach has received many accolades and awards, including a Leadership in Action Award from the University of Florida, and she is also a Robert Wood Johnson Foundation Future of Nursing Scholar alum. Her research has been funded by the National Institute on Aging, the National Heart, Lung, and Blood Institute, and the National Human Genome Research Institute. She is currently funded by a NHGRI Choices for Sickle Cell Reproductive Health Award and 2 NHLBI awards to investigate the contribution of biosocial factors in sickle cell disease and the association of a SNP in the vasopressin receptor 1A gene with pain and psychosocial variables. Dr. Roach, a warm welcome to UTHSC and to the GGI seminar series.
Thank you. That's one less slide that I have to have to do. Okay, so I'll get started. I'll have to screen share. Sorry about that. 1 second while I. Can you see the screen?
Yes, that looks good.
Are you seeing the same screen that you saw earlier?
Yes, the pain genetics and sickle cell disease with the red blood cells.
Perfect. Great, because it's not showing very well on my computer. Okay, sorry. Okay, so thank you for that warm introduction. I am excited and honored to be here with you all today. As Megan stated, my name is Keesha Roach, and I'm an assistant professor here in the College of Nursing at the University of Tennessee Health Science Center. Today, I would like to talk to you about pain genetics and sickle cell disease as it relates to my body of research, vasopressin, stress, and pain in sickle cell disease. I have no potential conflicts of interest to disclose, and I have a listing here of some of my grant funding over the years that got me to this point. And my career path, I'm just going to go quickly through this slide as we already covered this just now. So a little bit about my background. As you already know, I started off at Howard University and got my first degree in psychology from the University of Maryland. I started off as a training and research at the National Institutes of Health, the National Institute of Alcohol Abuse and Alcoholism, where we examine genomic approaches to understanding alcoholism and substance abuse in American Indians. I later worked on projects with a focus on molecular genetics and type 2 diabetes at the Howard Hughes Institute at the University of Chicago, the Study Center for Human Polymorphisms in Paris, and the Pasteur Institute in Lille, France. Upon return to the US, I felt that my research would be richer if I had a better understanding of the clinical side of the work I had been doing. So I applied to an accelerated BSN program and became a registered nurse, where I worked in a clinical setting for several years. And after witnessing patients suffering from vaso-occlusive crisis, pain, and sickle cell disease, I developed a strong interest in this disease state. And it became the focus of my doctoral training program at the University of Illinois, Chicago, where I obtained my PhD. Next, I completed the T32, as Megan already stated, at the University of Florida in the Pain Research Center for Pain Research and Intervention Center of Excellence, followed by a faculty position at University of Florida and now University of Tennessee Health Science Center. And the attraction to UTHSC was because of the strong history of sickle cell research in this region. And now I will have the ability to continue my program of research using omics as a tool to better understand the biopsychosocial factors in neuropathic and nociceptive pain. During today's presentation, I'll review the underlying pathophysiology and a brief history of sickle cell disease, types of sickle cell disease pain, genetic underpinnings of pain and sickle cell disease, and future directions. Sickle cell disease is a group of inherited blood disorders involving hemoglobin, which was first described in 1910, over 100 years ago. In many sub-Saharan African countries, pain and sickle cell disease are so intimately intertwined that there are tribal words to describe pain for the disease state that are onomatopoeic and have been spoken for hundreds of years before Hendrick described sickle cell disease in the Western literature. Recently, genetic variants revealed that the mutation for sickle cell disease had a single origin 7,300 years ago during the Holocene wet phase, which is primarily sub-Saharan West Africa. And this was work done by Schreiner and Rotini's group. Sickle cell disease is caused by a missense point mutation in the beta globin chain of hemoglobin, where the DNA nucleotide adenine replaces thymine, thus causing an amino acid change from glutamine to valine, resulting in crystallization of hemoglobin molecules when oxygen levels in the blood are low, leading to sickling of the red blood cells. People who have sickle cell disease have abnormal hemoglobin, called hemoglobin S, or sickle hemoglobin, in their red blood cells. Hemoglobin is a protein in the red blood cells that carries oxygen throughout the body. And people who have sickle cell disease inherit 2 abnormal hemoglobin genes, 1 from each parent. The types of sickle cell disease include SA, hemoglobin SA, which is a sickle trait, hemoglobin SS, which is full-blown sickle cell disease, hemoglobin beta O thalassemia and beta plus thalassemia, hemoglobin SC, SD, and SE. And there, of course, are a few others that are typically seen in Asian countries like the Middle East, for example. Sub-Saharan sickle cell is the most common and most often the most severe form of sickle cell disease. Hemoglobin SC and hemoglobin S beta thalassemia are 2 other common types of sickle cell disease, while hemoglobin SD and SC are much less common. Today, sickle cell disease is a global health issue. The prevalence of the disease is high throughout sub-Saharan Africa, the Middle Eastern basin, the Middle East, and India. There's an estimated prevalence of approximately 300,000 births per year worldwide, and it is most prevalent in Nigeria, the Democratic Republic of Congo, and India. In the US, sickle cell disease affects approximately 100,000 Americans. The majority of those affected are of African ancestry and occurs in about 1 out of every 365 Black or African American births, and 1 out of every 16,300 Hispanic American births. About 1 in 13 Black or African-American babies are born with sickle cell trait. And the estimated incidence for Hispanic Americans born with sickle cell trait was reported as about 6.9 cases per 1,000 births, but that information was limited as it only included 13 states. This slide represents sickle cell inheritance combinations for individuals without sickle cell disease or trait, individuals who have trait, and individuals who have sickle cell disease. So when one parent has sickle cell trait and one parent has a normal gene, each child has a 50% chance of having, or each birth, I should say, has a 50% chance of having trait and a 50% chance of being normal. And then for those where both parents have sickle cell trait, each birth has a 50% chance of having trait, a 25% chance of having sickle cell disease, and a 25% chance of being normal. For the babies who have parents who have this sickle cell disease and a normal parent, each of those offspring will be born with sickle cell trait. And for the individuals with where the parentage is sickle cell disease and sickle cell trait, for each birth, the child will have a 50% chance of having sickle cell trait and a 50% chance of having sickle cell disease. And if both parents have sickle cell disease, all of the children will be born with sickle cell disease. Sickle cell disease is a multi-system disorder. Every organ system in the body is impacted from an early age. People with sickle cell disease have the potential of being plagued with stroke, blindness, acute chest syndrome, heart attacks, renal failure, leg ulcers, avascular necrosis, liver damage, splenic sequestration, and chronic pain, just to name a few. Vaso-occlusive crises are characterized by episodic, recurrent, and unpredictable episodes of acute pain. Microvascular obstruction during vaso-occlusive crisis leads to an impaired oxygen supply to the periphery and ischemia, reperfusion injury, inflammation, oxidative stress, and endothelial dysfunction, all of which may perpetuate a noxious microenvironment leading to pain. Acute painful episodes due to vaso-occlusive crisis and its release of the mediators contributed to the inflammatory milieu that triggers the nociceptive activation. Whereas the chronic pain in patients may stem from edema, inflammations, ischemia, or nerve damage, and can be considered as neuropathic pain in nature. Neuropathic pain typically has two types. One due to tissue damage following vaso-occlusive crisis, and the second seems to be the consequence of persistent chronic pain. This affects major organs of the body, as I previously stated, brain, heart, lungs, kidney, peripheries, etc. This complex pathobiology of sickle cell disease suggests that pain may occur via nociceptive, inflammatory, and neuropathic mechanisms. Each pain state generates its own neurochemical signatures in the periphery and the central nervous system and requires different analgesic treatments. Nociceptive pain in patients with sickle cell disease had been thought to be primarily related to visceral and somatic tissue injuries subsequent to vaso-occlusion events. Emerging evidence from human and animal studies suggested that a component of sickle cell pain may be related to the neuropathic process. Significant knowledge has been obtained from studies of molecular and neurobiological mechanisms leading to and maintaining neuropathic pain. Some of the most promising evidence has implicated major roles in protein kinase C, also called PKC, and calcium calmodulin independent protein kinase 2, also called KMK2, and their interaction with N-methyl D-aspartate receptors, NMDA, and the transient receptor potential vilanoid 1, V1, which I'll use the abbreviations moving forward for these receptors. And so these are all involved in the development of neuropathic pain. The latest evidence suggests that these pathways are important for sickle cell pain as well. So coupled with animal studies of sickle cell disease, the researchers from our group began to elucidate the neurobiological mechanisms underlying pain in this disease state with the goal of gaining a better understanding and effect of the disease, of the pain state, along with effective therapies. So here we're showing the positive feed-forward loop with protein kinase C and CAMK2, as it may lead to the sustained activation resulting in neuropathic pain. So both PKC and CAMK2, which can be activated by the NMDA and TRPV1 via phosphorylation. So studies performed by our group at the University of Illinois examined patients with sickle cell disease who were given pain descriptors that included words that identified nociceptive and neuropathic pain. In a sample of 25 patients, 92% of the participants selected neuropathic words to describe their pain. This was novel at the time because pain was commonly thought to be nociceptive in sickle cell disease. It was not clear if the finding was due to the small sample size, so the study was repeated with 145 participants. And this time, the participants chose neuropathic words to describe their pain 91.7% of the time. So then the team took the evidence and designed studies in the transgenic mouse model using quantitative sensory testing, heat pain and mechanical pain stimuli. They found neuropathic pain in the Burke mouse. So a little bit of background about the Burke mouse. The Berkeley humanized mouse model, the mouse alpha and beta globin alleles have been knocked out, and human alpha and human sickle beta globin alleles have been inserted. And this mouse displays the pathophysiological changes observed in patients who have sickle cell anemia, including organ damage, hypoxia-induced cell sickling, and pain severity. So now we know that sickle pain can be nociceptive from tissue damage or neuropathic from nerve damage. And this study provided more evidence of the neuropathic pain in this population. Following studies leading to further evidence for neuropathic pain among adults with sickle cell disease revealed the need for adjuvant analgesic treatments for these patients. So based on preclinical studies, it was highly promising that trifluoroperazine would show efficacy in humans. Trifluoroperazine, also called stelazine, which is a common name, is a phenothiazine antipsychotic drug used to treat schizophrenia, anxiety, and other psychotic disorders. So the CAMK2 has a known role in neuropathic pain, and stelazine is a potent CAMK2 inhibitor. Researchers from our group wanted to determine stelazine's acute effects primarily on adverse events and secondarily on pain intensity reduction in adults who had sickle cell disease. So in a phase 1 open label study of 6 doses of trifluoroperazine, so those doses were 0.5, 1, 2, 5, 7.5, and 10 milligrams. We obtained 7 hourly and 24-hour repeated measures of adverse events, pain intensity, and supplemental opioid analgesics in 18 patients with sickle cell disease, each of whom received a single dose. Participants reported moderate to severe effects at 7.5. Moderate to severe sedative effects at 7.5 and 10 milligram doses respectively. 8 of the patients reported 50% reduction in chronic pain without severe sedation or supplemental opioid analgesics. And 1 of the participants had dystonia at 24.5 hours after the 10 milligram dose. The analgesic effects lasted for 24 hours in 3 of the participants, and the sedation was resolved with caffeine and dystonia was resolved with diphenhydramine. Adults with sickle cell disease experience minimal adverse effects at doses under 10 milligrams. In this molecular mechanism-driven translational study of stelazine showed promise as an analgesic drug that is worthy of further testing in a randomized controlled study in adults with sickle cell disease, starting at a dose of 1 milligram and repeated doses to determine long-term adverse and analgesic effects. So the participants in this study provided even more evidence for a molecular basis for neuropathic pain. And it's also given us hints for therapies for neuropathic pain. And that the dose from 1 to 7.5 milligrams is going to be effective for different individuals and the dosing could have genetic variability predictors in some of the individuals as well, as they may metabolize stelazine differently. Some may metabolize it different than others. There may be ultra metabolizers, normal metabolizers, and of course, low metabolizers. Some may only need to have 1 milligram dose, where others may need to have 7.5 milligrams to be able to get pain relief. There was one patient in this study of interest who had 38 acute care utilization visits in 1 year and actually spent 180 days in the hospital in 1 year. This participant got the best benefit from stelazine dose. So this is therapy based upon the molecular evidence, and it was carried back and forth between the bench and the bedside. Stelazine is expected to be most effective in chronic but not acute types of sickle cell disease pain. So we need to understand the genetic underpinnings of this neuropathic pain and vaso-occlusive pain. So far, this has all been about trying to understand these sickle cell pain types. Genomic studies focusing on pain are rarely studied in the sickle cell population. A significant barrier to adequately address the pain in sickle cell disease is the insufficient information about underlying mechanisms affecting the variable degree and types of pain experienced by these patients. Our group prioritized and examined the genotype and allele frequency of 115 SNPs from 49 candidate genes and 199 adult African American and pediatric patients of African origin who have sickle cell disease. Analyses were performed and compared with available data from public databases. As a result, genotype and allele frequencies of a number of SNPs were found to be different between our cohort and those from databases, and between the adult and the pediatric participants. We found that the genotype and allele frequencies for a number of SNPs were statistically different in our cohort, and the literature data for African Americans, and also between our UI sample of primarily African American adults and the Northwestern sample primarily of West African origin, more specifically majority Nigerian. As previously stated, sickle cell disease is a multifactorial disorder. There are multiple factors that contribute to the pain experience. Multiple biopsychosocial factors known to contribute to other pain conditions have not been studied in sickle cell disease. Multidisciplinary pain research will aid in opening the path to personalized pain medicine. So we reported on patients with sickle cell disease who participated in a study of associations between genetics and pain phenotypes. I chose the SNP for Arginine Vasopressin. It was a SNP number 108777969. I chose this one because of its relationship to and my interest in pain and its relationship to stress. This is where my years in the lab began to merge with my clinical experience. Specifically, from my clinical experience, I was able to postulate that the AVPR1A polymorphisms might help to explain some of the clinical observations in sickle cell pain, specifically since in patients with sickle cell disease, clinical factors such as pain and stress have been associated with increased healthcare utilization, but it is not known if the presence of this SNP plays a role in this observation. So from a previous exploratory analysis within our group of COMT gene, single nucleotide polymorphisms, and DRD3 SNPs by our group suggested possible contributions to pain-related acute care utilization in people who have sickle cell. Our aim was to extend the analysis to gene-spanning haplotypes of COMT SNPs and DRD3 SNPs to investigate possible associations with pain intensity and pain-related acute care utilization in a sickle cell cohort. Genotyping was conducted and clinical data were collected, including self-reported pain intensity using Pain Reported, which collected an average of current pain, least pain, and worst pain in a 24-hour period and reported out as API, average pain intensity. And medical record extracted pain-related information and acute care utilization for 130 patients. As a result, haplotype blocks were identified based on linkage disequilibria. COMT had 7 haplotype blocks and DRD3 had 8 haplotype blocks. Regression analysis tested for association between haplotypes and API and utilization with several significant findings. For COMT block 1, the AGG haplotype was associated with higher API compared to the reference that was statistically significant at 0.02, whereas the AAA haplotype was associated with higher utilization, significant also at 0.02. For DRD3, block 2, relative to the reference, the TCGC haplotype was associated with higher utilization, significant at 0.01. And for DRD3 block 4, the AGT haplotype was associated with higher acute pain index, significant at 0.04 as well, and utilization. In conclusion, we found DRD3 and COMT haplotypes associated with pain-related sickle cell disease features, suggesting that future studies should be performed. So it is important to consider, in addition, cis effects of the SNP alleles in evaluating genetic contributions to sickle cell disease pain and acute care utilization for pain. So just a little bit of background about the Arginine vasopressin. This is a 7-transmembrane domain G protein polypeptide that has been identified as being involved in many neurological functions, including aggression, bonding, sex behavior, autism, and schizophrenia. Clinically, it's related to vasoconstriction, salt and water retention, and behavioral phenotypes like stress. The single nucleotide
Keesha, I have a question for you. Could you go back a slide?
Okay.
I'm sorry, one more. The slide, yeah, there. So you've mentioned you had 4 haplotypes for COMT and 4 for the dopamine receptor. Is that right?
No, there were 7 for COMT and 8 for DRD3.
Okay. So how do we read this in terms of the haplotypes? In other words, was there a particular haplotype with a strong additive effect, or
So I do know that that most of these were due to an additive effect. And so this manuscript is actually in submission. It's not published yet.
Hey, Keesha, do you actually, this is Andy Griffith, do you actually have the data to show us?
I do have the data, but it was too much to put on a slide. The way that I had it, presented to the journal that I'm submitting to. I couldn't get it to fit with and let you still be able to see what was actually on the document. It would have been way too small. I tried.
Okay.
I guess what I should have done, and I apologize, I should have just written out the different blocks for CompT and DRD3 that had significant results and showed what the reference was and the haplotypes and then whether it was API, whether it was significant for API or utilization.
Yeah.
Yeah.
That's what I think we were looking for. Yep. Okay,
Yeah,
got
and
it.
that's what I was thinking about that as I was saying it. I was like, this demographics is not helpful. So I apologize. But if you would like to see this information, I can definitely share it with you afterwards.
Great. I see Athena has a question for you in the chat. Did you find sex differences in haplotypes? If not, do you think females experience more pain than men? Could hormones play a role in pain crises?
Hormones may, but I didn't look at that. And I do, in the next slide that I was going to talk about, the next few slides, I was going to bring in the sex effect because I am seeing it. I've seen it in some of my past work that I'm going to report on, and I'm seeing it in some of the current analysis that I'm doing now, and writing a manuscript for my AIM-1 paper. So I'll go ahead and move forward. Were there any more questions right now, for now?
Looks like you have one more, but maybe you can circle back to that after you talk about sex differences.
Oh, okay.
Comment.
Okay, so I'm going to finish up on this, just introducing a little bit more information about the Arginine vasopressin that I chose. And so basically this SNP is 108-77969, which is a part of the Arginine vasopressin, which is part of the gene. So the SNP of this gene is found in the promoter region of AVPR1A on chromosome 12. And the protein coded from this gene behaves as a receptor for arginine vasopressin and belongs to a family of G protein coupled receptors that include oxytocin. This family of G coupled receptors mediates cell proliferation and contraction, platelet aggregation, glycogenolysis, and coagulation factor release. And most recently, vasopressin was described as playing a critical role in the modulation of pain, and several studies have reported that AVP can regulate the pain process in the brain through mediation of the central.
Is anyone else having trouble hearing, or is it just me?
I hear.
Just you, Megan.
Okay, great, continue.
Okay. So this SNP has been reported that it plays a role in pain, in the pain process, in the brain through the mediation of central cholinergic and opioidergic systems. A three-way interaction had been reported by Mogul's group out of McGill between genetics, sex, and acute stress when observed in both, and it was also observed in both mice and humans. So for this study by his group, male volunteers reported stress in a model of capsaicin pain that differed based on genotype, and no effects of stress was reported, or no effects of the stress by genotype was reported in women. And findings from the mouse gene encoding AVP translates to humans in its role in pain and genotype interactions with sex and acute stress. So a little bit about the work that I did subsequent to that. So in my pilot study, we examined the relationships between this SNP and self-reported pain, stress, and acute care utilization. And so my study was frequencies of this SNP from pain-related candidate genes. They're available now in individuals with sickle cell disease. But one of those genes, as we already stated, that Arginine Vasopressin, has been associated with pain and disability in other populations. In patients with sickle cell disease, clinical factors such as pain and stress have been associated with the increased healthcare utilization, as I stated, but it's not known if this SNP played a role in this observation. So the purpose of this particular study was to explore the relationship between my SNP of interest and self-reported pain, stress, and acute care utilization events among patients with sickle cell disease. So this was a cross-sectional investigational study about patients. And the participants completed pain reported, which is a computerized measure. It's a computerized version actually of the McGill pain tool. And it's used to describe their pain experience. And they also contributed blood for genetic analysis. We extracted emergency department and acute care utilization data from the medical records, and 107 participants were in this study. Utilization events, let me make sure, Utilization events were defined as the number of admissions to the acute care center or the emergency department for pain control over a 12-month period. And that served as a marker for acute pain. And then for pain, we use pain reported, which is a well-validated, modified, computerized version of the McGill pain questionnaire that measures pain intensity, location, quality, and pattern, by which a single composite pain index score was calculated from the average pain intensity number, the number of pain sites, pain pattern, and the pain rating index total scores. And the proportional scores were averaged to generate a composite pain index on a range of 0 to 100. And the CPI, this is a new way, or was a new way at that time, of conceptualizing and scoring the McGill pain questionnaire as a patient-reported outcome using a single score. And when collected at a routine outpatient clinic visit, it had been shown to be a strong predictor of acute care utilization over subsequent year in adults with sickle cell disease, and it was replicated over a second subsequent year. So, and last but not least, for stress, we examine patients' reports of stress by reviewing free text descriptions of pain aggregators from pain reported. This information was coded independently by 2 researchers who created an indicator of stress. So we had stress or no stress mentioned as an aggravator of pain. And then we use this for data analysis. No report of stress as an aggravator of pain was coded as 0, and spontaneous reports of stress as an aggravator of pain was reported as 1. The researchers used a consensus process to determine the code when there was discrepancy, and ultimately it produced 100% agreement. The SNP genotype frequencies for this sample were 30% CC, no, 20% C, 28% CC, 41% CT, and 31% TT. And acute care utilization and stress as an aggravator of pain were significantly associated with this SNP at 0.02 and 0.002 respectively. The CT genotype had the highest mean utilization, and the CC genotype was associated with not citing stress as a pain aggravator. And then chronic pain was not associated with the SNP. So in the conclusion for this study was that the SNP is related to indicators of stress and acute pain, and further research is recommended for other measures of stress and acute pain. And this was the first study of ABPR1A among individuals with sickle cell disease that showed that individuals with the CT genotype had higher acute care utilization, whereas those with the CC genotype were less likely to spontaneously report stress as a pain aggravator. Sickle cell disease, as we know, is a genetic condition known for pain and is associated with stress and inconsistently associated with sex, two variables that other researchers have found to be associated with this SNP. So further research is warranted and should include pain, perceived stress, and other psychosocial measures in a larger sample with adequate distribution by age, sex, and ancestry. Some of the limitations of this study was that it did not formally measure stress. We derived a stress measure from individuals who had spontaneously reported stress as an aggravator of pain. The number of participants who would actually report stress as an aggravator of pain may be higher than what we observed in this sample if we had included specific stress self-reported questions. So, and also our eligibility criteria excluded patients who had not had recent acute care utilization. And it's unknown if the findings would differ had they been included. The sample size was also a limitation in the study, that it was exploratory, and the study was conducted based on an available sample of people who were participating in a larger study. And finally, it's unknown if findings from other diasporas within the sickle cell disease community would have the same outcomes, since our sample did not include individuals from Saudi Arabia, India, and other countries with a high prevalence of sickle cell disease. So for here, the problem is not only the need to replicate using validated stress measures, but also to understand the biological and psychological mechanisms of stress in sickle cell disease. This study has been funded, and I'm currently in the process of obtaining IRB approval here at UTHSC so that the grant might be transferred over. So the first thing that I will be doing, which is from a data set of 172 individuals, I'll perform statistical analysis to test for associations between the ABPR1A genotype and experimental pain, which will determine whether pain thresholds vary based on the ABPR1A genotype. So I hypothesized, I used quantitative sensory testing. So I hypothesized that thermal and mechanical pain thresholds will differ by genotype. Also, I want to test whether the relationship between sickle cell clinical pain and environmental stress is moderated by the RS108-77969 genotype. And my hypothesis is that the genotype will differ, that the genotype will influence the correlation between environmental stress and clinical pain. This work is also involved in recruiting and characterizing a new cohort of 50 adults. So I'll recruit from here, from the Memphis area, with sickle cell to study additional biologic effectors of the vasopressin system for mechanistic insight. And this is to test for the relationship of pain and stress phenotypes with ABPR1A gene expression and with promoter methylation. So to explore and also I want to explore the variability in arginine vasopressin, plasma concentration, and associations with pain and stress. And of course, this data will be used for future grant funding based on my findings. So digging a little deeper, as I stated previously, the SNP of interest is located in the promoter region of ABPR1A. In the middle is the next SNP that I analyzed, and the third SNP is shown on the right. All are in very strong linkage to disequilibrium. So the alleles for these travel together with the major phase indicated at the bottom. So the three major haplotypes present down here at the bottom of the page. The last SNP in the rather large 3' untranslated region and could have a regulatory effect. So like it might affect microRNA binding, for example. Each SNP could have an effect on the gene, even if it's silent substitution, and could have an even larger effect together. No one has shown the effect of this SNP, and it's unknown if this SNP is affecting the amount of gene expression. A clear understanding of the impact of the protein production here could help explain the pathways. The scientific premise of this indication of protein contributions to pain phenotypes and their interactions with psychosocial variables will result in greater understanding of sickle cell pain, which may lead to improved medical management. So for this, also from the cohort of 50 that I'll recruit here in this area, this study aims to determine the effect of environmental stress related pain and age on protein profile of vasopressin system in patients who have sickle cell disease. So we'll develop a network associated with the protein of interest, which will provide a direction of focus for further investigation. And this data hopefully will be used for future grant funding as well, the output from this. So the approach is that the participants will complete psychosocial questionnaires and provide blood samples for plasma. And the proteins will be identified and further analyzed to determine differentially expressed proteins. These aims will shed light on the role of the vasopressin system in sickle cell pain and may provide insight for future studies and prediction and treatment of pain in this population, particularly stress-related. The overarching goal of the proposed study is to characterize the role of the biological marker in the Arginine vasopressin system as it relates to sickle cell pain. So future directions, I have SNPs. There are many SNPs that I have that have been sequenced and some imputed from this sample and or from the previous sample where of the 115 and also from the 130. And I would like to collaborate with individuals who are interested in working with us to analyze this data and move the work forward for publications and future grants and collaborations. Additionally, I would like to include disease severity, inflammatory markers, stress, and ancestry to add to my toolkit. So these are my references. I would like to acknowledge my mentors to the point of being at the University of Florida. So I have not included Dr. Goodman on here, who is the best addition to my mentoring team ever. And also the patients at the University of Illinois Clinic, along with the University of Illinois and University of Florida, and of course the Price Center and the Center for Palliative Care Research and Education. And I would like to thank you. And also I would like to acknowledge, this is one of my favorite art pieces. This artwork is by Hertz Nazare. He's a sickle cell warrior who passed last week. And so I just wanted to kind of give him a shout out and acknowledgement as well. Thank you. Questions?
Thank you. That was great. And you know, it might be a good time to circle back to Ansley's question, since you were talking about the, you know, recruiting adults here. So I don't know if you can see the chat or not, but she was.
I'm going to stop my share. Well, no, I should be able to see the chat this way too. 1 second. Oh, I might have to stop the share.
Or Ansley can ask. There she is.
Hi, Ansley. I can see it now.
So one of the things that I was more of a comment really than a question, it was just thinking about, because you had a pretty broad age range of subjects, if you're going to include differences by biological sex, then you need to consider menopausal status when you were doing so. I don't know that in that pilot sample of 130 or something patients that you really have the sample size to be able to support that, but it's something to think about in the future, and perhaps some of the collaborators on the call, potential collaborators on the call, could help you in recruiting that larger sample size. That would be certainly an interesting effect. You know, we've all heard the effects of hormones, the hormone surge that occurs during childbirth and so forth. And of course, menopause is a time when hormones are way out of whack. And I wonder how that affects pain during a sickle cell crisis experience during menopause. That was just a comment.
Thank you. Yes, and there are many manuscripts about that, and I have not made that my focus. And to be honest with you, I don't think it'll be too much of a problem to add another question to at least start gathering that information and then start delving deeper over time. So thank you for that.
I will also add that within the College of Nursing, we have one of the leading experts on menopause care. And so she could certainly help you with that. She would be happy to.
Interesting. Thank you for sharing. So you'll have to slip me her names in the email.
Keesha, cool presentation. I have a quick question. Well, maybe not so quick. We'll see. So as you know, probably as well or maybe better than I do, because you do more human genetics than I do, candidate gene analysis of this type has come in for tremendous flack over the last, 15, 20 years, as people move to GWAS, and the false discovery rate on candidate gene, at least in the area I'm familiar with, schizophrenia. So I've been reading this book it's a fascinating book by Ken Kendler and Jonathan Flint. And it was written in 2010, which was sort of the low point for GWAS and candidate gene analysis. Some hilarious stories about the failures in schizophrenia. So that of course raises my, what few hackles I have on the top of my head immediately when I see a candidate gene analysis, and I get worried about it and false discoveries. So I think there's an expectation that you'll have to factor that in pretty seriously. But the mechanistic question really is, so you've got RS309, et cetera. You've analyzed it every which way you can with the population you have, and you were very forthright about some of the limitations. Do you think that promoter variant is acting through expression differences? And is there any evidence that those humans with this haplotype or this genotype have higher or lower expression? In other words, do you have an actual mechanistic model that high expression means higher pain sensitivity or lower pain sensitivity or interaction with other gene variants?
Right. So thank you for that question. That's a great question. So first, I want to respond to the part about looking at the various SNPs and taking that candidate gene approach. And I agree with you, that has been, it hasn't been feedback that I've gotten with submission of manuscripts, but it has come up at different meetings that I've attended when I'm talking to colleagues from across the country. So that really helped me to focus my work on where to go to next, which is why ultimately I would like to add additional omics, and that right now I'm adding proteomics to my toolkit and moving forward in that way and specifically I will be looking at high pain versus low pain, and what am I seeing in protein interactions for this particular SNP.
Cool. Yeah, Jeff, one of the impressive things about Jeff Mogul, who you mentioned, is that he's one of the rare individuals who's been able to balance animal model work with some very cool human genetics on pain sensitivity. The melanocortin 3 receptor story that was published in Science many years ago was a great example. He's got more recent, really high profile papers in it. In many, if not all of those, he plays both with animal models and human cohorts. And that's really a unique opportunity for you here at UTHSC. And let me share my screen briefly.
Yes, please.
So here is data for Arginine vasopressin receptor 1A in the hypothalamus. We have it for midbrain, et cetera. It does for 50 different genotypes. It doesn't have much variation, 1.5, but that's a good start. And then you can ask all kinds of questions. You can say, what does it map? Does it map cis or trans? But what's more fun is you can then go up here and say, what is the correlation to all published phenotypes that have ever been generated in the family? And that's about 10,000 quantitative traits. And you just push a button here and it'll calculate all the correlations for you. I won't bore you with the results, but this is kind of unique.
Not boring at all.
So, but
Wow.
those kind of, so Megan is an expert on this. We have lots of expertise. David is on this. He's an expert. Athena is an expert. Klaus is an expert. I'm just looking at everybody who's listening to your talk right now. Chelsea's done a lot of this, I think. Pam Watson, Paige Lemon is an expert on this. So there's a lot of people who could play with the data a bit and see whether they can find some mechanistic links for you.
Great. Well, that would be great. And I'm definitely looking forward to meeting people here and having conversations. I'm really glad to be a part of GGI. I don't know if we've made that completely official yet, but this is going to prove to just be an awesome learning experience for me and a platform for me to just bring my work forward. And I'm so glad to be here. Any other questions?
I guess I can ask a question. So do you think, so you showed a set of haplotypes, and I'm not exactly sure, what, ancestries were represented there, but do you think that by studying admixture in different populations, you might be able to uncover rare variants or different types of haplotypes that might be informative for sickle cell?
Yes, and that's the hope. And I have not completely moved in that direction yet. I wanted to get settled here first, but I've reached out to a few people just to get on their radar. And I don't know who's here at UT who does that, but right now I'm not in constant communication with Rick Kittles, but I'm at least on his radar. So that is something that I would like to do moving forward.
I think that's a great idea. There's so much admixture of West Eurasians and West Africans in America to not put obviously we know where that comes from. But that is an incredible signal, that admixture. You'd expect, for example, that if you have sickle cell and anemias and trait in individuals with higher or lower West Eurasian admixture, you might have a different spectrum of sickle cell trait or sickle cell disease. I mean, the disease is what's going to interest you more, but even the trait is kind of interesting in terms of hemoglobin carrying capacity at high elevation, things like that.
Yeah, and I actually have been thinking about ways to move forward in that direction. And I was supposed to go to Sierra Leone this coming December. And I actually had permission from my program officer, but that trip looks like it's not going to happen. So I've been reaching out to individuals here at University of Tennessee Health Science Center who might be doing research already in other countries like Nigeria or Ghana, where I can identify a sickle cell group. I know, for example, one of the largest sickle cell groups is in Ghana, but I don't have a contact. I don't know who to reach out to, and I've been trying to find someone. So hopefully I'll be able to do that.
So Keesha, Bob Davis works in Ghana, and we have a group, I think it's a center in our college, which you more than welcome to join. And they've been very active on multicultural global health. And a lot of them do work in Africa. And if you'd like, I can send you the contact information.
Okay, I would love that. I really would appreciate that a lot. So thank you.
You might even enjoy just, I think they have a seminar every month, and oftentimes they're about, projects involving global health, and a lot of them are in Africa. All of them that I've seen are in Africa, so that could be helpful.
Keesha, Claire Simpson in GGI also works very closely with him in the African cohort.
And she's part of that group.
Okay.
That will, so the beauty of admixed populations is the haplotype blocks are huge. So if you look at African Americans, there will be haplotype blocks that are like half a chromosome, or, just huge chunks. So now if you imagine a situation where you have two admixed individuals that are both, let's just speculate, they're 40% West Eurasian and 60% African ancestry, but they're unlucky enough their kids now have sickle cell. Now because of that West Eurasian admixture, you can look for the modifiers. So that's what I would say. If it was me doing this, I would say it's the admixture that makes this powerful. It makes it almost like a family study instead of a GWAS. So if you could find, let's say you found 1,000 individuals with sickle cell in the Memphis area with admixtures that ranged from, say, a low of 2 or 3%, which would be quite low, to a peak of maybe, it's going to be like 70 or 80%, right? And now if some of those, then you'd expect a very different spectrum depending on the extent to which modifiers influence pain sensitivity. So, we do these kinds of tricks in rodents all the time because we can do the breedings we want to do, and we intentionally mix it up that way.
Yeah, that's definitely a way that I would want to potentially go. I don't know how you said 1,000 there. I think that I looked at the numbers for Regional 1, for example, and Methodist, and I don't know if they quite reach 1,000, or they might be at 1,000, which you don't get everybody when you're collecting data. So hopefully I would be able to have strong findings with a smaller sample size.
I think that's possible. And later this month, we expect to have 9,007 genomes back from Regeneron, of which 44% will be African-American kids, of which I don't know how many will have sickle cell, but it'll probably be in the 50 to 100 range. And you could easily use the genotypes there and the exome sequence to find out what their admixture ratio was, and then see whether you just see any kind of correlation. You'd obviously have to contact them, which would be a little tricky. It's not a protocol that allows recontacting in a trivial way, obviously.
Oh, okay.
But that would be a fun thing to.
So this didn't come through like a registry?
No, it's basically waste blood that gets used for genotyping.
Okay. Thank you, though. There might be a way to pull it off.
Hey, Keesha, I just sent you, I actually didn't remember this, but tomorrow there's a symposium by this multicultural global health group, I guess in Mooney. And I sent you this. So let's
Is
see.
this the link? No, that's not.
Yeah.
I don't. Okay.
It might be, did you send it through the chat? I think, because also Ansley had a message for you in the chat too.
She said she had her-
I don't have a link. What I have is, well, maybe I can paste the registration in the link.
Is this the South African group?
This is a large group and it looks like a huge program. And I think you'll meet people from every corner of the globe if you're able to drop in. I know it's late notice, but.
Well, you never know. I may be able to. And it's, what times is it? 7 hours ahead of us time zone, or is it?
No, it's in, it'll be held in, let me take a look. I think it's going to be held in the Mooney building, which is that beautiful library that Dr. Brown is renovating. I think it's right next to Crow.
It's attached to Crow, and it's 8 to 4 tomorrow.
Yeah.
Okay.
And they include breakfast and lunch.
Okay. Well, that's good to know. And my schedule is open. And even if it wasn't for my work, it can always change. So thank you for sharing this.
Yeah.
Do you
cool.
know but you don't know the name of the group, right?
Oh, it's the Center for Multicultural and Global Health. It is their inaugural Multicultural and Global Health Symposium. The title of this one is Reimagining Multicultural and Global Health in the Age of COVID. There are just all sorts of people on the agenda. I'd say looks like there's about 20 something people on the agenda to speak, panels,
Wow.
research presentations, including Claire and our own Mike Witt, as well as many others. Probably a good group to plug into though.
Yeah, and it's in person, not virtual, right? Yeah, it's in the Mooney building. Yeah, so I guess maybe I'll meet Claire tomorrow.
It looks
both,
to,
right?
it looks
It's
to me
both, right,
like,
Andy?
sorry, Rob, I keep butting in.
Is it both? I guess the question is, I've spaced out. I don't know about this. Was it an open invitation?
I'm pretty sure anyone is welcome.
Can you put the link to the meeting? I would love to go.
It's in the chat box.
I see the link to the Zoom, but not to the meeting itself. Oh, I see. Maybe that'll work. Sorry.
I also sent a link, I sent the email to you, Ansley, and I think I tried to send it to Keesha. I'm going to have to step off for another meeting, but let me know if you have any trouble. I'd be delighted to try to, facilitate that. Thank you very much.
Patty just added the link. Thank you, Patty.
Yeah, terrific. Thanks, Keesha.
Thank you. Thank you all so much for joining me today.
Yeah, thank you. That was a great talk and great. As usual, I really like the discussion at the end.
Yes, I'm glad this is recorded.
All right, well, yeah, go ahead, Klaus.
Yeah, if I may. Keesha, this class, I don't have my video on, so you just hear me. I may have missed it. Do you know where this gene, this Arginine Vasopressin, is expressed? In which tissues?
Oh, yeah, it's in the platelets. I don't remember all of the tissues right off the top of my head, but I do know platelets, it's one of them.
All right, because I just looked at my data. I'm currently collecting blood from patients that are infected with influenza, not a huge collection, about 300 or something like that in the end. And we've been collecting in Memphis and also in Chapel Hill. And there's a good representation of Afro-Americans in there. And I just looked at it. It looks like it's expressed in the blood. So I only have the bulk expression levels. But what might be interesting, and I've so far I haven't done any detailed analysis, I also have the genotyping. So I don't know, we may perhaps be able to look at those SNPs or haplotypes in correlation with levels of expression of the gene in the blood of these people.
Okay.
So just keep it in mind, and we can get in touch sometime later.
How do I say your last name?
Klaus Schugart, Schugart. I put it in the chat.
Thank you.
Yeah, that
Megan,
was a,
can we be sure that I get the recording?
yes, for sure. In fact, I'll send you a recording after it completes downloading to the cloud for approval
Thank you.
before I post. Yeah, well, that was a great addition by Klaus at the end there. So if there's no more questions, thank you again, Keesha, that was great. And I hope everyone has a great weekend.
And
Thank
register
you.
for that class tomorrow. I just did, or presentation.
Okay,
So-
I'll go register now.
Yep. All right. See you.
Nice meeting everyone.
Nice to meet you too.
Bye.